Hi, I'm Susan, this is Hayley, and welcome back to Cancer Research Demystified. This episode is all about models of cancer used in the lab.
Okay, so during a previous video, and we'll link to it in the comments below in the little description, we talked about how we grow cancer cells in the lab. And we said in that video we would go into some more modern techniques, something a bit more complicated. So in this video we're going to do 3 different techniques, all which are state-of-the-art ways of growing cancer in the lab.
So primary cell culture is the first technique that we're going to discuss. Now, unlike cell lines, which we talked about in our previous video, these can only be kept alive for a short while. To describe what they actually are, we're going to go with a seasonal metaphor. It might not be Christmas for you anymore, but it still is for us. So if you look at this Christmas tree cartoon, hopefully coming up here, you can see that it's sort of our metaphor of a tumour. It's got lots of different cell types or decorations, all different complicated things going on in the body. Primary cell culture is where we use chemicals to dissolve away almost all of these decorations, just leaving one type on, or one cell type, that we can then keep alive in the lab just for a short while.
Okay, and then what we can do now is with those different cell types that we separated out, we can actually add them back on. Maybe 1, maybe 2, maybe a few different ones. And that is hard, and it's really complicated, but it's possible, and it's definitely what we're doing in the lab these days.
So the next technique is where things get a little bit more complicated again, and we call this organoids or tumouroids. So to explain this, we get a slightly more realistic looking tree. This is our department Christmas tree, so you can see it's a little bit closer to real life. In biology terms, we're adding in things like the molecules outside of the cancer cells, the different substrates that they interact with and the spaces around them. This still isn't quite as good as real life though. You can see this tree is still plastic. It's not a real one. It doesn't even have any tinsel, which in this case would be blood vessels. Yeah. And the fairy lights aren't even switched on, meaning it doesn't have a functioning immune system since it's outside the body.
But it is 3D.
Yes, it is 3D.
I can knock it over.
Don't knock it over.
Hi, so we've moved away from the Christmas tree and back into the labs. So these are the same places that we use for a normal primary cell culture in that other video, and we'll link to that so you can kind of understand how these would work. This is where we also do the more complicated cell cultures, and Susan's going to tell you about something she does for her project.
Right, so getting that little bit more realistic again from organoids, we have something that I use, and it's called gelatin sponge culture. So these are the gelatin sponges. They're actually the things that dentists put inside your mouth to soak up the blood when they're doing gross things to you, because they're these sponges that soak up liquid really, really well. That's what they do. So I use them to soak up liquid that has food in it for our cells to keep them alive. So I put them inside little wells of this plastic plate, and then I put the tissue on top of the sponge. So this is tissue that is straight out of a patient. I haven't dissociated it. It's like the full real Christmas tree, not a plastic one. It's the exact tumour, the exact way that it was in the body. We keep it alive for a few days, and then we can use that to test new drugs and therapies. It's pretty good, but again, although it does have some blood and some immune cells, it's not in a living body, so it doesn't have a blood system functioning and an immune system functioning. But it's as close as we're really going to get in a plastic environment.
So it can get a lot more complicated than that. You can actually have artificial structures that have shape to them, and we'll cover them in cells and try and grow things almost to the sort of level of an organ. We don't personally do this in the lab here, but we have colleagues in the university who do. So if you have any more questions about any of these new complicated ways of cell culture and how close we're getting to kind of reproducing
Reproducing,
the real
replicating,
life,
yeah.
So if you have any more questions, put them in the comments below and we'll see if we can answer them for you.
Okay, bye.